Here, we would like to present our recent research concerning the nanosuit, which can observe the living and wet biological samples by a scanning electron microscope. Please remind that biological samples contain about 80% of water, and a SEM needs high vacuum conditions. Humans are called vision animals. Because we can understand when we see something, we invented a telescope to see the far object, and the microscope to observe the close ones. From the life scientific point of view, we know the history of the observations using a microscope. When our ancestors invented light microscopes, they recognized that organisms are composed of cells. It was a big paradigm shift for our understandings. We are eager to see smaller objects. The historical senior scientists invented the electron microscope, which has more high resolution than the light microscope. The electron microscope made a paradigm shift to know the existence of organelles in a cell and viruses. Because of its high resolution ability, it is now an indispensable tool for many scientific fields including life science. When you use the electron microscope, you need the specimen put into the high vacuum condition, because the molecules of gases and water vapor absorb electrons. The pressure level in the electron microscope is almost the same as in space at the outside of the space station. Please imagine that you are released into the circumstances without a spacesuit. You would be choking and losing the water and air from your body in a moment. Therefore, it is easy to imagine the living organism is destroyed when you put them in the observing chamber. The larvae of Drosophila were destroyed completely. This is the main reason why we cannot observe living creatures as it is with an electron microscope. Therefore, our senior scientists improved the method to observe the dried specimen, which imitates the living creature. The photo shown on the right side is the result of the conventional method which all scientists are now using. To compare with the photo taken by the light microscope shown on the left side, I wonder whether the result of the conventional method reflects the real structure of living organisms. I have come to want to observe living organisms with a scanning electron microscope while they are still wet. In the early history of the electron microscope, Pease and others reported that they observe a living coleoptera in a SEM, and after the observation, it moves again under the atmospheric pressure. Therefore, we put several animals, such as Hydra, planaria, and larvae of mosquito in a SEM, without any traditional treatment. Many of them except the larvae of Drosophila, those structures were destroyed because of the high pressure. When we put the larvae of Drosophila into the vacuumed chamber in the electron microscope, we found they were moving. Why? As I showed you the crushed larvae in the former slide, but this one is moving. Why can they live in a harsh environment like space? How can their lives be protected in harsh circumstances? Then I came up with an idea. I could make up the nano-sized spacesuit. Drosophila larvae have sticky mucous membranes on their bodies. When I switched on the electron beam as soon as possible, we could see the movement of them, but when I switched on after 30 minutes to 60 minutes, we found the dried samples as shown on the former slide. When we observed the section of the red square area by transmission electron microscope, we could see the additional nano-sized thin film around the cuticle of this animal. This nano-thin film may act as a spacesuit. As a small conclusion, when we irradiate them by electron beam, we can make them alive in a harsh environment, and also when we irradiate them by plasma, which has the same role as an electron, we also can save their life. But if we keep them 30 minutes to an hour, the destroyed structure is observed. The electron or plasma irradiated sample had an additional nanomembrane, but without any irradiation, no additional membrane was observed. We now realize the polymerization by electron or plasma energy. This nano-sized membrane seems to play an important role to keep life in a harsh environment. Therefore, we could learn from nature to keep the lives of many animals. Nanosuit solution must be a liquid that mimics the Drosophila larva's mucus substances, which we call extracellular substances, ECS. We analyzed the extracellular substances, but in vain, because the mucus contained many different biological compounds, such as proteins, hydrocarbons, oils, etc. From a biomimetics point of view, we mimic the ECS artificially. I tried applying it to coat a mosquito larva's body, which is usually drying out in a vacuum as shown in the lower left figure. Then I put the artificial ECS on. As a result, it moved as usual like this slide in an electron microscope. They can survive and move in harsh conditions with electron beam bombing. And some of them have emerged to adults. We coined this functional membrane as the nanosuit. By using the electron beam or plasma irradiation with nanosuit solution, we found the nanosuit on the surface of the cuticle, and the fine structure of the mosquito larvae were ordered. However, without the nanosuit solution, the structure was completely destroyed and no membrane was observed. In order to check the transparency of the nanosuit, we made a transmission electron microscopy to measure the thickness of it. When the thickness of the nanosuit was less than 100 nanometers, we can see the fine structure of the body. When we covered the surface of the nanosuit with metal using the scattering method, we cannot find the animal's body, because the reflection or secondary electron has occurred at the surface of metals. And we confirmed the existence of the metal membrane above the nanosuit using a transmission electron microscope. The arrowheads indicate the nanosuit, and arrows indicate the metal surface on the nanosuit. We also observed some plants. This is a very famous tree in Japan, cherry blossom. It is a symbol of spring in our country. Because the petal of the flower has some waxes, we tried them in the electron microscope as it is. We can observe the fine structure. When we remove the wax gently from the chloroform solution and observed it in the electron microscope, the structure was immediately broken. The result shows that the petal of cherry blossom possesses the natural substance which can polymerize to make the nanosuit the same as the larvae of Drosophila. We compared the existence of nanosuit using by transmission electron microscope. We can see the nanosuit above the petal and no treatment with electron beam irradiated and washed by ethanol and electron beam irradiated ones. However, there was no nanosuit was observed no treatment without electron beam irradiated one and washed by chloroform and electron beam irradiated one. We can gather the wax using the solution washed petals by chloroform. With that solution, we can make a self-standing membrane made on the metal mesh shown in the figure below. We also developed the nanosuit method for medical research. For example, the nanosuit can apply to C tissues of mouse and human cultivated cells. The upper column shows the mouse peritoneum, and the low column shows the human fibroblast cells. The apparent differences were observed between the nanosuit and the conventional methods. In this experiment, we used a modified nanosuit solution which we coined as nanosuit type 3 for naked tissues and cells. This figure is the image that shows the tissues of human stomach cancer with the nanosuit. The upper part masked by violet color is the cancer area and the lower part is the normal tissue area. The front line is very important for the diagnosis. The time for this preparation needs only 5 minutes. This slide shows viruses in a cell in a wet condition. Small dots are viruses and the lower right part is a cell. You can see a lot of viruses around the cell and also on the soma of the cell. The cell possesses the long filament at the center of this figure. We also can observe exosomes as other biological particles in addition to viruses. Those photos show the exosomes with nanosuit and without nanosuit. Exosomes are lipid bilayer covered microvesicles shed by several tissues including solid tumors into body fluids, such as blood and urine. Exosome diagnosis has demonstrated the ability to extract exosomes from the blood and urine of patients with a variety of cancers. It is very useful not only for minimally invasive diagnostic methods but also for the discovery of cancers that are difficult to diagnose like pancreatic cancer. Many researchers trying to detect the origin of exosomes by key genes, but we are now trying to detect them by a direct morphological method using the nanosuit method in the electron microscope. Without nanosuit solution, the exosomes were disappeared during electron microscopic observation. We are now possible to combine light microscope and scanning electron microscope with the same medical samples. First we observe the normal pathological sample, and take a photo using light microscope. And then remove the cover glass, and apply a small amount of nanosuit type 2 solution. Then we can observe by a SEM. This slide shows the application of the nanosuit method to pathogens in paraffin sections. The figures at the colored column are stained by hematoxylin and eosin and were taken by a light microscope, and those at block and gray column are covered by nanosuit membrane and taken by a field emission scanning electron microscope. The top row indicates the lung infection with fungus, the second row is entamoeba histolytica infection of the colon. The third row is bacterial infection, and the last row is the cytomegalovirus infection of a kidney. The same area observed by a light microscope can be enlarged by a scanning electron microscope. Those pathogen slides are able to resave after the observation. Since the nanosuit method needs only few procedures comparing the conventional method, we tried to analyze elements that reflect from living organisms using energy dispersive X-ray spectrometry, EDS. In order to increase the sensitivity, we put 2 EDS in this electron microscope. Although it has been reported to produce crystals of calcium oxalate when the aloe plant was hurt, it does not know the details such as its localization and morphology. We used the nanosuit method to observe them. This is the morphology of the crystal of calcium oxalate and the conformation of calcium of it. Instead, when we use the conventional method, it was hard to observe the crystals because of the several procedures including the replacement of solutions before the observation. We can also use the EDS for nanosuit correlative light and electron method to the glass slide. The left column shows the figures taken by light microscope, and the rest of them are taken by EDS scanning electron microscope. For an example, please see the second and third rows showing the charcoal powder deposition in the lung. The light microscope could see the dark spots in the lung. EDS analysis shows the different metals in the different depositions. Those results indicate the patient possessed several different metals in the lung. Comparing with the conventional method and the nanosuit method, the treatment procedure of the nanosuit method is very simple. As a conclusion, we now observe the living and wet biological structures using the nanosuit method, which needs only a few minutes. We scientists, a few years ago, couldn't imagine that the living creatures were able to live under the electronic microscope. As we found the nanosuit method, we can discover the way to observe the in-life creatures. I really think the precise observation of living organisms is important for biology, biomimetics, and medical research. With our new findings, I hope we can find the road to making a new lifestyle or creating new products.